Hello. My name is Nathaniel Edisis, and today I'm talking about From Freezer to Findings, Oxford Nanopore Sequencing for Rare Disease Research. So I want to start by highlighting the diagnostic odyssey that patients and researchers go through to precisely and accurately diagnose rare diseases. In 2008, a baby was born with hypotonia, and a year later, a muscle biopsy diagnosed them with congenital fiber-type disproportion myopathy, or CFTD. CFTD is characterized by smaller type 1 skeletal muscle and is a monogenic disease of mixed inheritance across a variety of candidate genes that are not yet all fully characterized. For this reason, the family decided to enroll in a research study at Boston Childrenâ Hospital a year later, and then 5 years after that, clinical exome sequencing revealed a single paternal SNV in the ryanodine 1 receptor gene, one of the candidate genes. Because of clinical presentation, a dominant RYR1 mutation expression was ruled out, and because there was not a paternal, there's a paternal but not a maternal hit, or a pathogenic variant in a dominant candidate gene, the case was left unsolved for 9 years until this last year when Nanopore long-read whole genome sequencing confirmed not only that paternal SNV, but also a maternal 4-exon deletion in that same RYR1 gene, which we were able to confirm using clinical methods, ending this patient's diagnostic odyssey 16 years after it began. So this highlights the work we've been doing at Boston Childrenâs Hospital Beacon for Long-Read Sequencing project, led by Drs. Wendy Chung, Alan Beggs, and Piotr Sills. The goal is to sequence up to 1,000 participants with previous negative genetic sequencing to answer the research question, which is what is the added marginal utility of long-read whole genome sequencing over short-read exome or genome sequencing in a diverse collection of rare disease cohorts. As a technical member of the team who worked in the wet lab, this meant different diverse cohorts, meant different labs, different extraction methods, different storage methods, from DNA as old as 30 years old to extracted this last year, which leads to a question: how does DNA quality affect long-read whole genome nanopore sequencing? So far, we've sequenced over 200 families and returned the data with an average coverage of 28x and a median N50 of 14.5 kb. And of the 141 families analyzed to date, we have an added marginal utility, or the rate at which a family had no diagnostic finding in short-read exome or genome sequencing, and then a research finding using Nanopore long-read whole genome sequencing of 24%, with those findings being disproportionately found in the congenital myopathies and dystrophies cohort. And when we look at the per promethion flow cell output, we can see that samples that were sequenced after being stored in a freezer in all ages have an average sequencing depth a little over 30x, 33x, but those stored in a fridge had about a 50% decrease in sequencing output, or an average of 20x sequencing depth, which meant that we were much more likely to have to use a second flow cell to reach sufficient depth for data analysis, really changing the economy of nanopore sequencing for samples stored in a fridge. But does this affect the efficacy? And from what we can see, we cannot see a significant positive correlation between sample storage methods and our ability to make research findings. And though there are significantly higher rates of research findings in fridge and freezer of DNA of all ages, and then fridge or DNA stored in a fridge for over 10 years, that is largely because they are overwhelmingly made up of the congenital myopathies and dystrophies bucket. And so in conclusion, long-read whole genome nanopore sequencing is a relevant and effective rare disease research tool. DNA storage method impacts the economy, but not the efficacy of nanopore sequencing. And for that reason, rare disease researchers can use Nanopore long-read whole genome sequencing to go from freezer or even fridge to research findings. Thank you so much.